is working on three important crops. One is horticultural crops, which are very important for Australia, 50 million Australian dollar export value. Wheat is very important in Western Australia. More than 60 to 70% of the wheat of entire Australia is produced in Western Australia. And pulses crop, which are very important source of the protein. So our group is working on the crops which provide the carbohydrates, the proteins, and other important micronutrients. So these are the important crops we are working. In the area that what we are doing for wheat, we are improving wheat for heat tolerance, drought tolerance, frost, because in Australia we got all these issues and nutrient huge efficiency. In the area of pulses, including chickpea, pigeon pea, peanut, on which I used to work earlier, we are working on drought and also enhancing the protein content, herbicide tolerance, etc. So these are the important aspects we are trying to address. In the horticultural crops, recently we got a project from Hort Innovation Australia, which is having worth about $15 million, and in this project we are delivering that these issues or addressing the issues related to the enhancing fruit size, resistance to variety of pests and diseases. We are working on 5 important horticulture crops, including banana, papaya, pineapple, and custard apple and passion fruit. So these are some of the research going on in our group. And what we are doing these things by using different genomics technology, including genome sequencing and analysis, where we developed the expertise during last 20 years. I will discuss some of those areas later. And for understanding the genome, you need to have very good tools and knowledge, and for that we use the computational approaches. And by combining these things, we try to understand the trait, the molecular basis of different traits. We use the genome engineering approaches, including the gene editing. And another important thing is that when we have these products, in context of India, we were working with the national program in Africa, but now in Australia, we work with the industry, so that these products, they go to the industry and then this can go to the society. And lastly, in my role as the international chair in agriculture and food security, we are connecting Murdoch University with international agriculture with the different countries or so. So I think this is some of the things which we are doing. So this was like an introduction. And what we are doing, that for different addressing these different stresses, either diseases, enhancing productivity, pest resistance, we are using the crop breeding approaches, because breeding is the core, genetics and breeding, which helps to develop the varieties. But then they are not the only approach. You need to have the integration of the different other disciplines, including physiology, pathology, entomology, and different kind of approaches. So they need to be integrated. So these are very important approaches that we need to work together. Now, from the perspective of the genomics, and we have been working on this aspect of the genomics, so I think in the context of genomics, as we said, that if you would like to address any trait, you need to know the genes that are controlling for that. Now, to understand that one, you need to have those genes in your hand, and for that, the basic thing is that you need to sequence the genome. And you know that well, that all the genes, etc., they are in the form of the DNA or the chromosome or so. So, what I will say that, fortunately, I have been really lucky guy that I have been evolved with this different genomics technology. Way back when I was doing my PhD in Prof. P.K. Gupta lab, and then also now in ICRISAT, and now in Murdoch, and then we will discuss some of these things in the future what we would like to do. So let's go back to the past. And when we did not have the genome sequencing technology advanced, many of us are aware about the Watson and Crick model, etc. of the DNA. But now in the plants, and if you would like to use that in your breeding program, then there was that evolution of the technology, which we say molecular markers, and they started in 1980s. Community around the world, they started to develop the genetic map, and this was the first map in 1980s, and they started from the human being, and this was a marker technology called restriction fragment length polymorphism. So by breaking those different segments and you combining the genetics, you can create those genetic maps. So this was the start of those development of those markers also. I'm talking about past, and then you had different range of molecular markers, including rapid, randomly amplified polymorphic DNA, and then we also had simple sequence repeat microsatellite markers, and this was the time that I started to do the research in the laboratory of Professor P.K. Gupta, where we were working on wheat, and I was involved in development of SSR or microsatellite markers. And these are the smaller repeat units in the genome. And what we do that we take them and we try to see in the different varieties or different individuals of a given species, understand polymorphism and then associate with the different traits. And then we have different technology including amplified fragment length polymorphism. And these are some of those things which I was doing way back in 95 or so. So we started to working on those technology. I'm not talking about this past, even for the sequencing. When I was working on my PhD, we did not have the automatic sequencer. We used this even manual sequencer at that time. And then we evolved, and even during my PhD, we had the automatic sequencer. So you take those DNA clone, put them in the machine, and the sequencer. So this was a very tedious process, but we were successful to do many things. And when I moved to Germany, even at that time point, we did not have those sequencing technology that much advanced in the plant side. So what they were doing that instead of sequencing entire DNA fragment or so, they were breaking those things in the smaller fragment, and instead of going to the entire DNA, you just target the expressed portion of the genome, and then when we were targeting those sequencing or we were sequencing, we used the word express sequence tag, so ESTs, and by using these ESTs, you could get some idea about the genome. So these were the things that they were moving, but then the huge boost came to this plant genomics from the perspective of the human genomics, and way back in 1990 when they embarked on a big project, and they thought at that time point that basically human diseases are a very important thing. Everybody would like to address them, and especially cancer and all these things. And I know that several of you are working, including our old friend Prof. Samirdas, all these. So, like in this direction, if you can understand the molecular basis of those traits or like those things, then you can really address those issues. So in 1990 they started, but the whole genome sequencing for human took around 13 years, and it costed about 3 billion US dollar. So this was big discovery kind of thing in the humankind, and many people they compare this kind of discovery when man landed on the moon. And I think that this genome was announced in the White House with the heads of the different governments of United States, UK, and many other places. Anyway, so now when they did this sequencing, and then next thing is that can we afford this kind of cost and that much time every time? Probably not. And then different communities started to think, and then they, because of these things, they challenged the scientific community. Can we reach a stage that we can sequence the genome not in the 13 years but just 30 minutes, not in the $3 billion, just in $1,000 per genome? And with this, to achieve or to win this race, there were many advances in the sequencing technology, etc. came, and then large number of genomes started to be sequenced. In the case of plants, in 2000, the Arabidopsis genome was sequenced for the first time, which was very smaller genome, and only 125 megabase pair. We had the 5 chromosomes, and the 2000, this genome was sequenced. And then at that time point, as I said, I was in Germany between the 2000 and 2005. In 2005, in terms of the crops, rice genome was sequenced, and this genome size was about 420 megabase pair, but they sequenced only 389 megabase per genome. So this was when we started to have this evolution of the technology in the plant side or in the agricultural side. When we were there, and we always thought from the translational perspective how you can use the genomics in the breeding of the crop improvement. And in the same year when this paper was published, together with my boss Andreas Runner and another collaborator from Cornell University, Mark Sorens, they had this 10th anniversary issue of the Trends in Plant Science. They invited us to discuss these advances and how they can be helpful in the breeding program. So we gave this concept called genomics assisted breeding, and how genomics can help the breeding program. And after that, this became very routinely used term. If you go to many conferences, people use genomics assisted breeding, etc. Anyway, so here we gave the concept of how you can use the genomics research in the breeding program. Now when I moved to ICRISAT, and we were working on the crops like chickpea, pigeon pea, and peanut, many of these crops used to be called orphan crops, because we did not have many genomic resources in these crops. If you see at international level, wheat, maize, rice, soybean, these are some key crops which are given a lot of funding, etc. But some of these crops which are grown by smallholder farmers, then you don't have those technology. But hence, to the evolution of the sequencing technology, the genomics technology, that initially they had these manual slabs, but then as many companies came in the picture, sequencing cost kept on going down and then throughput kept on increasing. So I think this was really big boost for the sequencing technology, but now during the last few years, range of the sequencing platforms they are there, and now you can sequence any individual, any organ relatively less time and the less cost. So this was something about that past, that how we were doing these different kind of things in sequencing. Now I would like to discuss something for last 10 years or so, that where we are, what we have been doing, even would like to highlight some of the examples from our own research. So when I moved to ICRISAT in 2005, we did not have, as I said, the sequencing technology, but then I was trained in Germany. So we started a genomics center at ICRISAT, and when I started, now we kept on evolving with these advances in the technology. So as a founder director for the center, we use this center for decoding the genomes of several crops. Before that, India participated in some genome sequencing consortium, but from the country we did not lead any sequencing program. But this was one of the key, that while staying in India, while working in India, we led the genome sequencing program for several crops. Anyway, we also celebrated the 10th anniversary of this center, and we had an honor to have Professor Bajaj Hussain, Professor Ali sir, and also Anwar. You may remember that picture, sir. Yeah, so although we could not have the other people, but nevertheless, we always remember you, and we look forward to host you at other places as well. Anyway, and now when I moved to the Murdoch University, we already have a center, but now in this center we are bringing new machines, and which are the state of the art. And recently, this new machine will be coming in the month of January. T7, in just one day, you can generate 7 gigabits per data. So this is very much fast forward looking approach. Anyway, so by using these different sequencing technologies, so machines can generate the data, but then they cannot give you the idea which gene and what. So for that, you need to develop the expertise. You need to have computational tools. You need to have not just computer program, but you need to develop the expertise to understand the data, to understand or interpretation of the data, and with using these things, we decoded the genomes of several crops, including pigeon pea, and this was published in 2012. Chickpea, pulse millet, and all these genomes we led from India, and they were published in all these good journals. And of course, we had a lot of partnership from the different organizations around the world. But when I was at ICRISAT, we sequenced the genomes for more than 12 different crops. And this itself is a big thing. Otherwise, many labs, they spend several years to sequence just one genome. And here, we delivered the genome sequences for more than a dozen crops. So I think this was one key thing. And second is that at that time point when we were developing these sequencing or genome assemblies, they were not very perfect, because at that time point I started to use this technology in 2007 onwards. But then with the time, the technology evolved, and now with this new technology like high-C technology, and this is called 3D genome sequencing, and now you can sequence the DNA of the entire chromosome together, and this is called the chromosome-led genome assembly. So you do not need to have the different context. So for the whole chromosome, you can have that the genome assembly. So these are advances. And right now, if you would like to see, because this is the time that now large number of plant genomes have been sequenced, and you can see large number of these things, and this list keeps on increasing every year. As I gave the example of Arabidopsis, things kept on moving, but now if you go, large number of genomes are sequenced, many of them, they have been contributed from our research group as well. After that, if you would like to ask the question that if you would like to develop, for instance, drought-tolerant chickpea variety, whether just having one chickpea genome will be helpful? Answer is probably not. Because if you have just sequenced one genome, and if this is not a drought-tolerant variety, you will not find the gene for the drought tolerance in your genome assembly, right? So, here for instance, if you sequence cultivar 1, cultivar 2, cultivar 3, they may have some unique genes which will now, if you sequence cultivar 1, now cultivar 3 got some unique genes. So these genes will not be present in cultivar 1. So we get the concept that well, we need to keep on sequencing large number of individuals, and once we have the sequencing data from these different individuals, you can develop the pan genome. So basically you have the genes for the entire species. So you would like to catalog as much genes as you can, and based on these things we enhance our concept further, and we say not only in a given species you need to go to the related species, and then you can develop the super genome. So based on these things we started to sequence large number of individuals. So like in the case of chickpea, we sequence more than 430 chickpea lines. In the case of pigeon pea, we sequence more than 300 pigeon pea lines. Sequencing is one thing, but now as I said, so after sequencing, what kind of value you can add, that's more important. So for instance, can you identify the genes associated with the days to maturity, days to flowering, harvest index, yield under drought and heat stresses. So these are the key things that how we can use the genomics technology to understand the genes for those traits which can enhance the crop productivity. And then we did this another very ambitious project where we just not sequenced few hundred, but then we sequenced 3,000 chickpea lines. And this was the first plant genome sequencing projects at the international level where so many genomes were sequenced. And this paper not only that from the perspective of the journal that published in the Nature, but most important thing is many international media houses in addition to India, they publish the stories or interviews from us, including New York Times, Economist, BBC, and many other papers also. So basically this was that kind of research that you can have the possibility now to sequence large number of individuals and how they are going to make the impact on the humankind. So these are the ways that we are moving ahead. I will not go in the technical details, but I just would like to mention that we are sequencing more than 3,366 genomes. We developed the pan genomes of chickpea, and in that, so we developed this pan genome, and now we have an idea that how many genes in the case of chickpea we got. So for instance, one genome did not have this more than, so these 1,600 genes, but now when we got the pan genome, we found the additional new genes. I will not go in the technical aspect, but by using these data, we were able to identify the center of origin, domestication, migration route, that how chickpea was migrated from its center of origin, which was infertile crescent, and then this kept on moving to the different parts of the world. Anyway, to make long story short, what we have been doing these whole genome sequencing projects in many crops, and by doing these things now for helping the breeding program, we were developing marker genotyping platform. The idea is that as a breeder, when they make the crosses, they generate thousands of the lines, put in the field, and then try to see that which line is good, right? But then sometimes if you are working on drought, in that particular year you got rainfall, then you cannot select the line for the drought tolerance. But if you got the genes for the drought tolerance, then after creating these populations, you can screen those populations in the laboratory with the genes. You do not need to depend on those natural conditions, etc. So now the breeders, they would like to screen the larger population in such a manner that you can have in the less cost. And with this objective, we developed a different kind of marker genotyping platform. Second is phenotyping. So if you are interested in grain yield, if you are interested in drought or heat, you need to screen your population for those different traits. And I think the good thing is that nowadays that you have a possibility to do aerial phenotyping, ground phenotyping, so you can generate large scale phenotyping data on all these things together. And at the present also when we were there, we developed a different kind of platform, and by using for instance these kind of things, you can measure the roots, you can measure the yield, you can do a lot of the field work. So by combining all these things data together, we were able, so we have used these different kind of approaches which are called genome-wide resources mapping, a lot of other stuff. And by doing these things, we were able to map large number of traits, including for drought, heat, etc. in these specific chickpea, groundnut, and pigeon pea. So this was the beauty of using all these different traits. So we sequenced the genomes, we understood the genes associated with the different traits. Now the next question is how you can develop a better variety. And from this perspective, we use this method called translational research. And I would just like to highlight the thing that here there are different kind of approaches, including genomics assisted breeding, marker assisted selection, marker assisted backcrossing, etc. These are some new approaches. What we did last few years, we used these approaches and we developed several lines. And when you develop the superior lines, they go for the multi-location trials in India and other countries. And if these lines they give the higher yield, other good trade, then they are released as the improved variety. And I'm very happy to mention that the several people, like from India, in the case of chickpea, many breeders, they were involved in our projects and they used several of those varieties, and especially for in the case of chickpea, because drought is a key thing. Can you develop the varieties with the higher yield under the rain-fed conditions and through the genomics assisted breeding approaches? So we took several of these projects as a part of the Bill and Melinda Gates Foundation, and I'm so pleased to mention that during last few years, large number of lines they have been released or are being released in India, in Tanzania, Kenya, and Ethiopia. And this was the first variety called Jelly 2, and this was released in Ethiopia, which was having 18% higher yield over the existing varieties. The same thing in India in 2019, Pusa Chickpea 10216, Super Nigiri 1, released by University of Agriculture Science in Harvard, Dr. Mannu, who was a traditional breeder. And this variety became the susceptible for the fusion wilt, the Nigri variety, but we put through the genomics, and these are the molecular breeding lines, and the variety he calls it Super Nigri, gives higher yield. Same thing, Pusa Manav. So these different varieties were in the 2019 and 20 and the 2021 also. For the drought tolerance, Pusa Chickpea 4005, Pusa Chickpea Vijay, fusion wilt resistance, and now even this year in 2022, through IRI, Pusa Chickpea Siam has been released, and even just 2-3 days back, this was a big news item in the media, and this was the variety Pusa JG16, drought tolerant and high yielding variety. So even in chickpea alone, by using these efforts, we were able to deliver more than 10 improved varieties which are having the yield more than 20%, and they are the different disease resistant, etc. Same thing in the case of pigeon pea. Some of the state agriculture universities breeding program, they have been developing these varieties, and this was for the fusion of wilt resistance. And in the case of peanut or groundnut, if you know that one, there are the oil composition, because this is the oil seed ground. You got the palmitic acid, linoleic acid, and oleic acid in the groundnut oil. Linoleic acid is not good for the health because this is having polysaturated fatty acid. In United States, you got the varieties which are having more than 90% oleic acid. So what we did, we changed those metabolic pathways and we developed the lines which are having more than 80 to 85% oleic acid. And in India, they released the first set of the variety Girnar 4 and Girnar 5, and they were the high oleic groundnut varieties. And Honorable Prime Minister Modi also dedicated or included these two varieties as a part of the 17 biofortified crop varieties on the anniversary, 75th anniversary of FAO, so that to dedicate these varieties to the nation. So this was for the nutrition aspect. Same thing in the case of groundnut or peanut, we had these molecular breeding lines for the foliar disease resistance, and not only in India, several of these lines, they showed really good performance in Ghana and Nigeria and other countries. So because at ICRISAT, we used to work in the different countries, so we were able to deliver several of these varieties in the different countries, and also some of these varieties in Andhra Pradesh and Telangana state also, so we had a lot of these different varieties. Now the last thing is that by using these varieties, so you can develop, you can release those varieties. Thing is, can you help the farming community, the smallholder farmers, to make more money? And we believe that you can develop these varieties, but they do not reach to the farmers in the real time. So even these varieties are existing with the research organism. And this is the key thing that we need to strengthen the seed system. And even so that we want that these varieties should be reaching to farmers in the real time. If farmers even get those varieties, but if they are not having the ideal agronomic packages, then they also do not produce more. And if they sometimes they produce more, and if they are not connected to the market, then their market crash down and they cannot make more money. So we believe that you need to have seed system, you need to have these agronomic packages, you need to address the issue of the post-harvest losses, and you need to connect the farmers to the market, and you need to have the value addition. Then probably farmers can get more money, and then also society can be benefited with this high value crops also. With this objective, when I was working as a global program director for the Grain Legume program, we run one project from the Bill and Melinda Gates Foundation, and this project was run for about 12 years. This was the total amount was $67 million. I had a privilege to run this project in 13 different countries in Africa, 2 countries in Asia, India and Bangladesh, and we were working on 6 different crops. And by using these different technologies, we produced, when I say we, so not Rajeev, Varshney, but many organizations, ICRISAT, CIAT, IITA, and 15 different national partners. So I provided the leadership. And they delivered more than 260 improved varieties, produced 500,000 ton seeds, and all these success stories, they were documented either in the form of the books or in the research journal, etc. And we were so happy that based on this work that this project helped 5 million smallholder farmer families, and for that work, ICRIS had got that Africa Food Prize for our impact in African countries for this native crops, and this is coming through the genomics, breeding, seed system, and multidisciplinary approaches. So this is some of these things that how you can use the genomics research not for just publication on the paper, but also delivering the improved variety to the smallholder farmers. So now you can ask the question. So I was talking about that what we are doing now in the genomics project, etc. What next? And next means just in few years, right? In next 5 years or something. We believe that if we need to have this food secure world, then we need to go fast forward breeding. And fast forward breeding means that you need to not using these technical or conventional approaches, but even in the molecular approaches, you need to go to the next level. And for that, we have provided this framework, and we say that when you got large scale sequencing data, you can go for the haplotype based breeding, and you can predict the phenotype of a line based on the genome sequencing data. So this is called the genomic prediction. These are the approaches. Genome editing you might have heard nowadays, and we need to combine these things together with the speed breeding, and then you can develop these varieties in fast forward manner. Here I would like to give an example that when you got the sequencing data of large number of individuals, you got the genes, you can identify the haplotypes, and then you ask the question, if your current varieties, they are having good haplotypes or bad haplotypes. If not, then you would like to progress the good haplotypes, you would like to develop the superior variety. For this purpose, when we were working here, we generated the phenotypic data for more than 3,000 chickpea lines at 6 different locations, 2 different years. We had the sequencing data, we combined these things together, and by doing these things, we found several haplotypes, several of these lines. And when we had Prime Minister Modi, which ticket we sent, and we had a possibility to take him to the field, and I don't think that this is working. Okay. Anyway, so this is not working. Otherwise, this was the video that he was going and then visiting those chickpea feeders. Anyway, and then we have some other approaches, so I will not go into details, but these are the other genomic selection approach where you can develop the varieties in the faster manner. Speed breeding is another approach that here you can take 4 to 6 generations in 1 year, and you can make the breeding process faster. So this is that way that now next few years people can continue. But now if you would like to see from the genomics perspective that in future that what is going to happen, of course we cannot predict the future, right? But at least we can have some idea. One thing is that, and as I said earlier, that if you see from the genome sequencing technology, so if when we were in 1990, we had just very simple genome sequence. At that time point, crops like wheat, which is the hexaploid 16 gigabase per genome, it was impossible that we could sequence the genome like in the case of groundnut also even 2 gigabase per, we did not have the possibility. But based on these technological advances, from 1990 to 2010 years, we were able to sequence the rice genome, and then we were even in the next few years, we were able to sequence even from our lab. In the case of groundnut, we were able to sequence 2.2 gigabase per genome, and nowadays there are lots of different technology, even they are having like size of the USB kind of thing, so you can sequence these genomes in much more faster manner. And one thing which is moving ahead now, that earlier we were sequencing the DNA of the entire plant. Nowadays people are talking to sequence single cell, so not even the tissue etc. You need to go at the single cell. You need to understand the temporal expression. You need to have the spatial expression, and those technology at the single cell level, you have the possibility. Now at Murdoch also we are establishing these platforms, and the approach is called histomics, so spatial and temporal omics approaches. So it will be possible to sequence the genomes at the single cell level and the large number of individuals. The other thing is the systems biology approach, and this approach basically brings all the different disciplines together because genes do not work in silo, so you need to have these integrated approaches. And this is the other way that if you would like to, because sometimes you study some traits in the lab, but they are not translatable in the field. Why? Because we have just understood some genes, not everything together, and this is from our lab that now what we are trying to do, we already work in some crops where we put this transcriptomics, proteomics, metabolomics, and epigenomics data together, and nowadays this is the possibility to use the artificial intelligence and machine learning approaches, and then based on these things, you can define a new set of the genotypes which need to be used for making the crosses, etc. Nowadays, synthetic biology is also getting a lot of momentum, and now the thing is that always people would like to see whether you can synthesize the new genomes in the lab. I think people have not reached to that lab stage, but at least there are several organisms that engage in this kind of process, and they have been successful to synthesize the genes of the entire bacteria, 10,752 genes. But the question is that by doing these things, can you create a new version? At this stage not, but we never know that what is going to happen in the future. Many people, they are going in this direction for creating those healthy level unicellular things so that they can address the issue of the biofuel, for instance. So there are several companies involved in these kind of things. The other thing is, people are talking that well, we can sequence the genome, so basically you can read the genome, right? Nowadays we talk about the gene editing, so you can edit the genome. Question is, can you write the new genome? So I think this is the another thing. We do not know what is going to happen, but these are the possibilities exist, because few years back, even when I was a student, we all know about this synthetic animal, etc. We will talk about later. The other thing is that during last few years, if you go 10,000 years back, we had a different kind of biodiversity. Right now, you are having a different kind of biodiversity. And people are talking that yes, we can create some of these things from the old time, and you can bring these bioabundance in the new time. So these are all very exciting things. These are not the movies or fancy things, but these are all scientific things based on the data. And I think this is going to happen. And synthetics, we have discussed a lot. I will not go in the detail. But I also had a visit this Edinburgh where they had this dolly, the sheep, where they created. But of course, we know that fate of this thing. We also know some of the issues of the human gene editing. So of course, there are some ethical issues as well. I'm not talking that we need to keep on moving by using science. We need to see the balance that where humankind should stay. But at least I can tell that you have lots of opportunities. Even nowadays, people are talking that instead of having this electricity, etc., can you create the plants in such a manner that in the drawing room you can have the light, so you don't need to have the light. So I think that a lot of stuff can happen through these genomics approaches. So this is, in my opinion, that I think these are some speculation. Some of them may be successful, but you may have many more things which we cannot even imagine right now. So I think the take-home message is that past of the genomics has been great, present is exciting, and future is going to have several new possibilities that even we cannot think right now. In addition to generating knowledge, and I'm talking from agriculture, but the same thing goes in the other side, in human health or animal or other areas, I believe that we need to focus on the translational genomics as well, so that we can deliver the better crops. And I always believe in this approach that alone we cannot do everything, but together we can do something, and partnership across the different organisms, across the different disciplines, across different groups is very, very much required. And I think that by doing this thing, we can enhance the R&D and contribute to develop the high quality products from the agriculture perspective or from inside, and so this should be available to the different. Lastly, in Perth, next year we are organizing PAG Australia, so in the case if some of you are interested, please plan to come. We will be very happy to extend all my help and support to you. And those of you who are working on wheat, we will be hosting International Wheat Congress in 2024 as well. So with these words, thank you very much for your kind attention, and I will